Okay, welcome everybody to the podcast on Chapter 11, dealing with the cardiovascular system. Today, what we're going to do is talk about the heart and how circulation operates. So, the cardiovascular system is a way to deliver oxygen and nutrients to living tissue so that they can generate sufficient ATP in order to perform processes critical to remaining alive. So, without a constant supply of blood, tissues would very quickly die because they would lack the oxygen and nutrients necessary to make the ATP that allows them to survive. Cardiovascular system is made up of a closed system, including the heart and the blood vessels. Closed means that the blood that travels through this system never leaves the confines of the heart and the associated vessels. It does not bathe the organs and the tissues. Instead, what happens is that gas exchange occurs at the very tiniest vessels in the system, which are known as the capillaries. In other organisms, they have what are called an open circulatory system, where the blood actually, in fact, bathes some of the other vital organs, and gas exchange happens uh, more directly. But in higher organisms, such as human beings, we have what we know as a closed circulatory system. The heart is a blood pump, and blood vessels allow the blood to circulate to all parts of the body. The functions of the cardiovascular system are to deliver oxygen and nutrients to cells and tissues, and to remove carbon dioxide and waste products from those cells and tissues. And if we didn't do that, we wouldn't be able to live very long, because obviously without nutrients and oxygen you can't make the ATP you need to survive. But more importantly, if you don't remove the waste products that build up as a result of metabolism, things like carbon dioxide and urea, that comes primarily from the breakdown of protein, what would happen eventually is that conditions around the cell would change to the point where the cell wouldn't be able to maintain membrane potential, and eventually electrical activity would shut down. That would end up finally killing the cell. Along with that, you'd also see enzymes begin to denature and their chemical reactions begin to cease. So this is an extremely important system to have developed over the course of evolution, this idea that we have the ability to transport these vital components to cells that need them in order to live. And really, it was a problem that cropped up in evolution as organisms became more complicated. Early on in the life history of this planet, organisms were fairly simple. They were what we would term single-celled organisms, which simply exchange oxygen and carbon dioxide directly with their environment, and the environment was generally uh, a liquid environment. But then, as organisms began to become more complicated, they began to evolve tissues. We had a problem before us in that we had to deliver oxygen and nutrients to tissues that might be far removed from intimate contact with the environment, and that's where the cardiovascular system provided a great advantage for those more complex organisms who were, because of their complexity, more able to survive in a variety of environments. So let's start talking about the blood pump, which is the heart. It's located in the thorax between the lungs and in the inferior mediastinum. It has a pointed apex that's directed toward the left hip, and the base points towards your right shoulder, and it's approximately the size of your fist. And you can see that it also lies right behind your sternum, which is one of the toughest bones in the body, and it's enclosed by the rib cage. And so this is another excellent design for the human body. This allows the heart a significant amount of protection. It is one of our most vital organs, and so it's nice that it is enclosed inside this bony cage. You'll also notice that it's very, very close to the lungs. The heart and the lungs work together in order to put oxygen into the blood. We have to pump it to the lungs from the heart, and then it has to flow back from the lungs to the heart, and that distance is short enough that this can be done very efficiently. If the heart and the lungs were far removed from each other, then we'd have a difficult time oxygenating the blood effectively. You can see here in the anterior view that we are not symmetrical in the thoracic cavity. The heart tends to lean left of midline. Again, there's the base and there's the apex. This is the diaphragm right here. And if we look at a transverse section, this would be the front of the body, this would be the back of the body, and you can again see the heart sitting right behind the sternum and right between the lungs. If we look at an anterior view, you can see that the heart takes up space on the left side of the thoracic cavity, and as a result, to compensate for that, the left lung has, number one, fewer lobes, only 2 here, as opposed to 3 in the right lung, and it also has a little notch called the cardiac notch to make some space for the heart as it takes up that extra volume inside that left portion of the thoracic cavity. As a result, turns out that the right lung does most of the ventilation, while the left lung is essentially there to help out. You can also see that the heart is enclosed in a double-layered sac called the pericardial sac. Inside that sac is a slippery fluid called pericardial fluid, and this allows the heart to beat without experiencing a lot of resistance, which would result in inflammation and scarring of the heart every time that it moved, which would be approximately once a second. So just like the other organs in the body, the heart has a very nice lube job, provided again by the visceral pericardium, which actually clings directly to the heart muscle itself, and the parietal pericardium, which surrounds the chamber inside which the heart sits, and between them, of course, is the pericardial fluid. While we're at it, notice that the lungs have exactly the same setup. They have a membrane that surrounds them called the pleural membrane, the visceral pleura clinging directly to the lungs, and the parietal pleura clinging to the inside walls of the chest cavity. There's the apex of the heart right above the diaphragm, and the base of the heart to which the great vessels attach. And if we zoom in on the heart a little bit more closely, we can see these great vessels. This is the pulmonary trunk, which branches directly off of the right ventricle. It is the gateway that the blood uses to get to the lungs. These are the pulmonary arteries. The blood flows through the left and the right pulmonary artery to the left and right lungs respectively. It then picks up oxygen in the lungs as we breathe, and then the oxygenated blood flows back to the heart through these pulmonary veins. You can see the left pulmonary veins and the right pulmonary veins there. The blood enters a chamber called the left atrium. As that chamber begins to fill with blood and the pressure forces the valve between the left atrium and the left ventricle open, what will happen is that the blood will spill from this upper chamber into the lower chamber, the left ventricle down here, and then the left ventricle will squeeze. It will then shut the doorway between the atria and the ventricle, and as the pressure builds, eventually it'll open the doorway to this big vessel here, the aorta, which is the gateway that oxygenated blood takes to the rest of the body. Then what will happen in the rest of the body is that the oxygen will be delivered to the tissues that need it for metabolism, as well as the nutrients in the blood, and the blood will in turn pick up carbon dioxide and waste products. It'll change color from a bright to a dull red, and then it'll eventually return to the heart through the inferior and superior vena cava. Notice here that these great vessels have a blue color to them because of the low oxygen content of the blood that's in them. These 2 great vessels will drain into the right atrium, which will then eject the blood into the right ventricle. The right ventricle will then squeeze. The doorway between the right atrium and the right ventricle will close, and then the pressure on the valve to the pulmonary trunk, called the pulmonary semilunar valve, will force it open. The blood will then head out to the lungs, where it'll pick up oxygen, and then the whole cycle will repeat itself. You can also see part of the circulation to the heart muscle itself. If you look here, you can see the left and the right coronary arteries that branch right off the base of the aorta, and they form a network of vessels that supply blood to the heart muscle proper. And because the heart is a living organ, just like the rest of our vital organs, it requires a constant supply of oxygen and nutrient-rich blood. The blood that's drained from the heart muscle actually enters the right atrium through a hole in the chamber. The blood drains from the sinus on the back of the heart directly into the right atrium. It's called the coronary sinus, along with the blood that comes from areas above the heart through the superior vena cava and below the heart in the inferior vena cava. If we close in on the pericardium, we note that it's a double-walled sac. The fibrous pericardium is very loose and superficial. It's basically connective tissue, allows the sac to remain attached to the tissue around it. The serous membrane is deep to the fibrous pericardium, and it's made up of two layers. The visceral pericardium, which you can see here is right next to the heart, okay, also known as the epicardial layer, and the parietal pericardium, which is the outside layer that lines the inner surface of the pericardial sac, and then between them, in the pericardial cavity, lies the pericardial fluid. It's basically a serous fluid, and it performs a lubricating function as the heart moves. So the heart's inside this double-walled sac all the time as it beats. You can see here also the layers of the heart wall. If we work from the outer layer to the inner layer, the first layer we come upon is the epicardium. Again, this is part of the pericardial sac responsible for secreting the serous fluid that lubricates the heart. The myocardium is made up of cardiac muscle cells joined end to end by intercalated discs. Remember that cardiac muscle tissue also has striations in it, as does skeletal muscle, which are indicative of the overlapping pattern of actin and myosin that we see in the sarcomere, which is the contractile unit of the muscle cell. And then finally, we see the endocardium, which is essentially an endothelial layer, which is capable of repelling the foreign elements of blood so that we don't inappropriately initiate the clotting cascade. So it's kind of slip and slickery on the inside. And it also provides a protective layer between the blood that goes into the heart and the other layers of the heart wall. Okay, the only way that we want to deliver blood actually to the heart muscle proper is through that coronary circulation that we talked about earlier. Okay, so you can see all these different layers: the epicardium, the myocardium, the endocardium, and this is the actual heart chamber right here. And then if we take a coronal section through the heart, what we see are the four chambers of the heart, the two upper chambers, the atria, which essentially receive the blood as a result of gravity draining the blood into them. The right atria receives blood from the entire body that's been depleted of its oxygen and nutrients through the superior and inferior vena cava, as well as from the coronary sinus, which drains again directly into the right atrium, and the left atria, which receives oxygenated blood from the lungs through the left and right pulmonary veins, okay, draining directly into this chamber. As these chambers fill with blood, what happens is that the pressure builds up. On the right side of the heart, eventually what that'll do is force this valve known as the tricuspid to open, where the blood will then flow from the upper chamber into the lower chamber. You can see the direction of blood flow here indicated by the black arrow. Actually, let's use a yellow arrow instead. It's easier to see. So this is the direction of the blood flow, okay, from upper to lower chambers. On the left side of the heart, the left atrium eventually builds up sufficient pressure to force the bicuspid valve open. And again, the names for these valves come from the number of flaps they have. There's 3 flaps in the tricuspid valve on the right side and 2 flaps in the bicuspid on the left. And then what will happen is that the ventricles will begin to exert their pressure on the blood that's in this chamber. It'll force these valves shut, okay, and at the same time as the pressure eventually builds, these two valves here, the pulmonary semilunar and the aortic semilunar, which is right there, will be forced open, and then the blood will flow to the lungs, okay, in the case of the pulmonary trunk, okay, that's to the lungs, and to the body in the case of the aorta, okay, so you can see the blood flow here. And what I want you to keep in mind here is that the way that the heart beats is just in the way that I've described it. The two upper chambers will fill with blood and then squeeze. Then as the two lower chambers begin to fill with blood, eventually they'll begin to squeeze, forcing these atrioventricular valves shut and these semilunar valves open, and then the blood will flow to the lungs in the case of the right ventricle and to the rest of the body in the case of the left ventricle. Okay, so this is why the ventricles have the nickname the discharging chambers, because they are the ones that actually send the blood out of the heart and to its respective destination. Now, a couple things I want you to note about the discharging chambers, the lower chambers here. Note how thick the wall of the left ventricle is, okay, compared to the walls of the right ventricle. This is the left ventricle right here. I'll trace it for you, okay. That's all left ventricle, all right. And then this is the right ventricle over here, and I'll outline that in blue. Okay, this is the right ventricle here. Okay, the reason that the right ventricle doesn't need to be as strong as the left ventricle does is because it doesn't have as much resistance to work against, since it only has to pump blood to the lungs and back, whereas the left ventricle has to pump blood to the entire body, hence the much thicker wall and the much more power that's present in this particular chamber. Okay. So again, note the flow of blood through the heart. The heart is always going to have some blood in all four chambers. It's just that the amount of blood will vary depending on whether that chamber is squeezing, where we say it's in systole, or whether it's relaxing, where we say it's in diastole. And then while we're at it, let's draw a line indicating how the blood is flowing back into the heart. We'll use white here. Okay, so the blood's flowing from here into the right ventricle. Okay, same thing here. And the blood is flowing from here into the left, or right atrium, I mean, and from here into the left atrium as indicated by these white arrows here. Okay, so you can see the blood entering the chamber as it comes back. All right. And here we go. Just like that. So systole when the chamber is contracting, diastole when the chamber is relaxing. Okay. The heart has several septa in it that separate the four chambers. The interventricular septum separates the two discharging chambers, which are the ventricles, and the interatrial septum separates the two atria, which are the receiving chambers. And you can see here, by taking a transverse section through the heart, again, the relative difference in thickness of the walls of the two chambers. Notice that the left ventricle has an extremely thick wall, while the right ventricle looks basically like a pouch coming off the left ventricle, again reflecting the fact that it doesn't have nearly as much work to do compared to the left ventricle. Also keep in mind, this is very important, that when we're talking about blood flowing through the heart, that it's always equal volumes that are discharged from each chamber in a simultaneous fashion. So the volume of blood that flows from the right atria into the right ventricle is the same as the volume of blood that flows from the left atrium into the left ventricle. By the same token, the volume of blood ejected by the right ventricle into the lungs is the same as the volume of blood ejected by the left ventricle into the body via the aorta. Always in sync. Okay. In the systemic circulation, blood flows from the left side of the heart through the body tissues and then back to the right side of the heart, whereas in the pulmonary circulation, blood flows from the right side of the heart to the lungs and then back to the left side of the heart. And so you can see here the pulmonary circuit, which is extremely short, okay, and the systemic circuit, which by comparison is extremely long. And the difference in the length of blood vessels that the blood has to go compared uh, between the pulmonary and the systemic circuit accounts for the difference in resistance that the heart has to overcome in order to pump the blood. Because the distance is so short between the right side of the heart and the lungs, we don't have to produce as much power to get the blood there and back. Whereas on the left side, we have to produce considerable power in order to push the blood around the entire body. Notice again that in this chart, the red blood indicates blood that's oxygenated. That means that the hemoglobin has bound to the oxygen almost completely. It's completely saturated. Whereas in the deoxygenated blood, the hemoglobin has considerably less oxygen bound to it and a lot more carbon dioxide, and hence it has this dull red appearance which looks blue when you look at it through the wall of a vein. Okay, so this is just showing you how the blood flows through the heart and back. You can see a little bit more clearly here the blood entering the right atrium through the superior and inferior vena cava, as well as from the coronary sinus, and then being pumped into the right ventricle. The right ventricle pumps it into the pulmonary trunk, which is then going to take it to the lungs via the pulmonary arteries. The oxygenated blood is then going to flow back to the heart through the pulmonary veins, and then as the left atrium fills, eventually it'll push the blood through the bicuspid valve into the left ventricle. The left ventricle will then squeeze. The bicuspid valve will then shut. The aortic semilunar valve will open, and the blood will flow out to the rest of the body. Okay, and that's why the left side of the heart, the lower chamber here, the left ventricle, has to be the most powerful of all four of the chambers in order to do that job effectively. Blood flow is only in one direction in a normally functioning heart. There are four valves. The atrioventricular valves are found between the atria and the ventricles. The bicuspid valve is found on the left side of the heart between the left atrium and left ventricle, and the tricuspid valve is found on the right side of the heart between the right atrium and the right ventricle. The pulmonary semilunar valve is found at the base of the pulmonary trunk, which is the gateway to the lungs, and the aortic semilunar valve is found at the base of the aorta, which is the gateway to the rest of the body. And if we look at the way these valves operate, they open and shut according to the differences in pressure that are on either side of the valve. They're anchored in place by special tendons called chordae tendineae that are often nicknamed your heartstrings. They open the heart during relaxation, and they're closed when the ventricles are in systole. And so you can see here in the cardiac cycle that we start with blood returning to the atria. This puts pressure against the AV valves, and they're forced open because the pressure is higher in the receiving chambers at this point than it is in the discharging chambers. As the ventricles fill, the AV flaps hang inside the ventricles. The atria will then contract and expel the rest of the blood into the ventricle. The next thing that will happen is that the ventricle will take over and begin to exert force, and as they contract, what will happen is that this back pressure against these two AV valves will force them closed so that the blood can't go back into the atria. And then as the pressure continues to build, what will happen eventually is that we will pop open these pulmonary and aortic semilunar valves. So notice here that these valves, these AV valves, are held in place by the chordae tendineae. If the chordae tendineae were to rupture, then what could eventually happen is that the valve could prolapse back into the previous chamber, resulting in a gurgling sound when you listen to the heartbeat through a stethoscope. And depending on how severe this valve defect is, it may have to be surgically corrected. But normally what happens is that these tendons keep these valves from everting, from flipping back in the other direction as the heart exerts force on the liquid that's in its chamber. Again, Very important to note that the volume of blood in this chamber is the same as the volume of blood in that chamber with regard to the amount that's going to be moved upon systole. Okay, the right side and the left side of the heart always have to keep up with each other. As the heart continues to build pressure, eventually what happens is that the pulmonary and aortic semilunar valves will open, and then the blood will flow into the great vessels. From the right ventricle, it'll flow into the pulmonary trunk, and from there into the pulmonary arteries. And from the left ventricle, it'll flow into the aorta, and from there to the entire body. And then as the ventricles relax, what will happen is the blood will actually back up briefly and bounce off of these semilunar valves. And that's a sound that you can hear in a stethoscope as part of the heartbeat. Okay, but the blood will not flow back into the ventricle because, again, these valves only open from the ventricles into their respective great vessels. Okay, so this is what ensures the one-way flow of blood through the heart. If we didn't have this, then the heart wouldn't be nearly as effective a pump because the blood would constantly flow back in the opposite direction owing to the difference in pressure. This ensures that the blood will enter the heart in one fashion and will leave the heart only via the pulmonary trunk and aorta.
Ventricular contraction causes the atrioventricular valves to close, which signals the beginning of ventricular systole. The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open. When the ventricles relax and their pressures drop, blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole. Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles. This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling.
The blood in the heart chambers does not nourish the myocardium. The heart has its own nourishing circulatory system consisting of coronary arteries which branch off the base of the aorta to supply the heart muscle with oxygenated blood. You can see them here and here. Okay, this is the right coronary artery here, and this will be the left coronary artery. The cardiac veins drain the myocardium of blood. The coronary sinus is a large vein on the back side of the heart that receives blood from all these cardiac veins, and the blood empties into the right atrium through the coronary sinus. Okay, so again here we're looking at the front of the heart, and you can see here part of the coronary circulation. Some of the other major branches include the anterior interventricular artery. Okay, you could see here the right coronary artery dipping under the right auricle, okay, and giving off other arteries such as the marginal artery, and then over here the left coronary artery branching into the circumflex artery, which will wrap around to the back side of the heart and give branches off to the posterior myocardium as well. So the whole heart muscle has got a very, very rich blood supply in order for it to do its work. You can see here the associated great vessels with the heart. The arteries include the aorta, which leaves the left ventricle, and the pulmonary arteries, which leave the right ventricle. The veins include the superior and inferior vena cava, which empty into the right atrium, and the pulmonary veins, which enter into the left atrium, bringing oxygenated blood back from the lungs to the heart. The superior and inferior vena cava will dump blood into the right atrium. The superior vena cava has an easier job because it's working with gravity in order to drain blood from areas above the heart, whereas the inferior vena cava has to work against gravity. And so one of the features that we're going to examine a little bit later on in this chapter is the fact that one of the things that separates arteries from veins, in addition to the direction of blood flow, arteries always carrying blood away from the heart and veins always carrying blood back to the heart, is the fact that veins also have valves in them that ensure one-way flow. And so this is part of what allows us to get venous return back to the heart, is the fact that we move around and the skeletal muscle, as it squeezes on the veins, forces the blood up through these valves in a single direction, and eventually all the way back through the inferior vena cava, back into the right atrium of the heart. This is why it's not advisable to try and stand perfectly still for extended periods of time, because the lack of blood flow back to the heart will result in an insufficient cardiac output to maintain consciousness, and you'll pass out. So anyone with military training probably knows that you have to at least squeeze those muscles in the legs and not lock your knees in order for you not to pass out. From the right atrium through the tricuspid valve, the blood travels to the right ventricle. From the right ventricle, the blood leaves the heart as it passes through the pulmonary semilunar valve into the pulmonary trunk. The pulmonary trunk then splits into the right and left pulmonary arteries that lead directly to the lungs. Oxygen is then picked up in the lungs and carbon dioxide is dropped off. And the reason that that happens is because of the differences in concentration of those gases in the lungs. If we're looking here at the lungs, there's our lungs right there, okay? And in the lungs, what we know is that in the air that we breathe, that the oxygen concentration, the O2 concentration, is very high, okay? And the CO2 concentration in the lungs is very low, okay? And so what happens in the lungs is that oxygen is going to load into the blood by diffusion, while carbon dioxide is going to diffuse out of the blood that's coming to the lungs from the heart, also by diffusion. And then the oxygenated blood will flow back to the left atrium of the heart through the pulmonary veins. The oxygen-rich blood then returns to the heart through the four pulmonary veins. The blood enters the left atrium, traveling through the bicuspid valve into the left ventricle. From the left ventricle, the blood leaves the heart through the aortic semilunar valve, and the aorta takes it to the rest of the body. Okay, so that's the entire trip. So if we trace it, let's trace it with a yellow arrow here so that we see the whole trip. So here is the blood coming back to the heart, okay, from areas above and below it. Sorry, here, okay. Blood then passes through here to here, out here, okay. Goes from here to the lungs. This is where it picks up oxygen. Then it's going to flow back to the lungs through the pulmonary veins. From there we're going to enter from the left atrium to the left ventricle. From the left ventricle we're going to head through the aortic semilunar valve to the aorta, and from there we're going to flow to the tissues of the body that need this in order to live. They need that oxygen, they need those nutrients. Okay? So that's the whole circuit right there. Now in order for the heart to beat as a functioning pump, it has to be electrically coordinated. The reason for that is that heart muscle cells have a property that's called autorhythmicity, where if we were to take a heart muscle cell out of the heart and put it under a microscope and give it oxygen and nutrients, and what we would observe is that it would beat all by itself. And this is a property that's called autorhythmicity. It's present in every heart muscle cell. But the thing is that not every heart muscle cell individually is going to beat at the same speed. Some of them will beat fast, some of them will beat more slowly. And that's not going to be an effective blood pump, because if all the heart muscle cells are beating to their own rhythm, what will happen is that the heart will essentially just quiver. And that's not going to be efficient enough movement to get the blood to flow through the heart and out the discharging chambers through the semilunar valves. So what we have to do is we have to coordinate this contractile activity, and the heart does that through what it calls a conduction system that has several parts to it. Okay, it has an SA node, which we think of as the heart's pacemaker, the AV node, which is the electrical gateway to the ventricles. We have here the AV bundle, also known as the bundle of His, that carries the electrical signals to the bundle branches, which carry the electrical signal down to the apex of the heart down here, and then the Purkinje fibers, which carry the electrical signal to the papillary muscles and up the walls of the ventricles, and then the whole thing will repeat again. The heart muscle cells will contract without nerve impulses in a regular continuous way, but not in a coordinated fashion. Special tissues set the pace of the heart. The sinoatrial node, known as the pacemaker, is in the right atrium, while the atrioventricular node is at the junction of the atria and the ventricles. You can see it right there. The atrioventricular bundle is in the interventricular septum. The bundle branches are in the interventricular septum as well, and finally the Purkinje fibers radiate up the walls of the ventricle. And what you're seeing with this pumping action is the way that the signal actually spreads. So following the dark arrows here, what happens is that the contraction signal is initiated by the SA node of the heart, the sinoatrial node, and it spreads across the walls of the right and left ventricle relatively quickly. The next thing that will happen is that the signal will reach the atrioventricular node, where it will pause shortly. So this sequential stimulation is important, because the first thing we want to happen is to force what we call atrial systole in the upper chamber, so that we can squeeze the rest of the blood out of the upper chambers into each of the ventricles. What happens as the atria squeeze is that the electrical signal is waiting here at the AV node for the blood to fill these lower ventricles. The atria, once they finish contracting, will then begin to enter diastole. What will happen next is that the AV node will take that electrical signal and pass it through the AV bundle to the bundle branches, and then down to the Purkinje fibers. And what will happen next is that the ventricles will squeeze and eject the blood into the aorta in the case of the left ventricle, and into the pulmonary trunk in the case of the right ventricle. Okay, so it's critical that we time this properly. We have to hold the electrical signal up for a short period of time to give these lower chambers time to completely fill as the atria go into systole, and it's only at that point that we allow the ventricles to squeeze, that force closing these atrioventricular valves and opening these semilunar valves, which are going to again allow the blood to flow to the lungs in the case of the pulmonary trunk, and to the rest of the body in the case of the aorta. Okay, so that's the way the signal spreads. Now, what I'll show you next is a little video that sort of illustrates how the electrical signal spreads across the heart, and also the type of electrical readout you get as that signal moves across the heart. So what you're going to see is a picture of the heart as it goes through systole and diastole, and you'll also see an EKG readout that will illustrate how the electrical signal moves through the heart as those electrical events spread, followed by the mechanical events that they induce. Okay, so stay tuned for that here in just a second.
Action potentials originate in the sinoatrial node and travel across the wall of the atrium from the sinoatrial node to the atrioventricular node. Action potentials pass slowly through the atrioventricular node to give the atria time to contract. They then pass rapidly along the atrioventricular bundle, which extends from the atrioventricular node through the fibrous skeleton into the interventricular septum. The atrioventricular bundle divides into right and left bundle branches, and action potentials descend rapidly to the apex of each ventricle along the bundle branches. Action potentials are carried by the Purkinje fibers from the bundle branches to the ventricular walls. The rapid conduction from the atrioventricular bundle to the ends of the Purkinje fibers allows the ventricular muscle cells to contract in unison, providing a strong contraction.
Okay, so as you can see, contraction is initiated by the sinoatrial node. Sequential stimulation occurs in all the other autorhythmic cells at the same time, and this forces the cardiac muscle depolarization to proceed in one direction, only from the atria towards the ventricles. Once the SA node starts the heartbeat, the impulse is going to spread to the AV node. The atria will then contract. At the AV node, the impulse passes through the AV bundle, then the bundle branches, then to the Purkinje fibers. The blood is then ejected from the ventricles and the aorta and pulmonary trunk as the ventricles contract. And so again, notice the wave that spreads across the walls of the atria, the pause at the AV node, heading down the interventricular septum through the atrioventricular bundle and the bundle branches, and then up the Purkinje fibers where we stimulate the walls of the ventricles, which then contract. And just as you saw in the repeating EKG, and also in the video that talked about the cardiac conduction cycle, it's important that the timing of this electrical signal not change significantly, because if it did, the ventricles might squeeze too early or too late. Okay, the atria might not squeeze at all in the event that we don't have any signal coming from the SA node. For instance, there's all sorts of possibilities that could interfere with the normal electrical signal that spreads across the heart and coordinates its pumping activity. Now, sometimes homeostatic imbalance can interfere with the normal functioning of the heart. A heart block results in a damaged AV node, and this releases them from the control of the SA node. The result is that you have a slower heart rate as the ventricles assume what we call an alternate rhythm. Okay, what happens is that the AV node begins to take over and doesn't pay attention to the stimulation that's coming from the heart's normal pacemaker. Ischemia is a lack of adequate oxygen supply to heart muscle, while fibrillation is a rapid, uncoordinated shuttering of the heart muscle, which results essentially in no blood being pumped from the heart. We can have fibrillation in the upper chambers, where it's called AFib, and this is a dangerous but not lethal condition, because the ventricles will still be capable of pumping blood throughout the body, but ventricular fibrillation is an immediate medical emergency, because when the ventricles aren't squeezing, then no blood is going to head through the pulmonary trunk to the lungs, or out through the aorta and to the body. And so you have approximately 5 minutes from the time that happens to start the heart pumping again in its normal rhythm. Tachycardia is a heart rate over 100 beats per minute, while bradycardia is a rate that's less than 60 beats per minute. What you're looking at here is just a look at the cardiac cycle and the heart sounds that are produced. Remember that when we begin the cardiac cycle, the atria contract simultaneously. They then relax, and then the ventricles contract. Remember that the definition of the chamber squeezing is systole, while the definition of the chamber relaxing is termed diastole. Okay, so it starts out atrial systole, then atrial diastole, then ventricular systole, then ventricular diastole. Okay, so if we wanted to put the signal in with that cycle, the signal is basically a depolarization of the walls of the heart. And so if we begin at the SA node, that would be a depolarization signal that would spread across the walls of the atria. That would be followed by atrial systole, and then repolarizing of the atria, followed by their relaxation at the same time, depolarization of the ventricles, followed by ventricular systole, and then repolarization of the ventricles, followed by ventricular diastole. Okay, so depolarize, squeeze, repolarize, relax in the upper chambers, then depolarize, squeeze, repolarize, relax in the lower chambers, over and over again. The cardiac cycle are the events of one complete heartbeat. In mid to late diastole, the pressure in the heart is low because the volume of the chambers is high. Blood flows from other areas passively into the atria, into the ventricles, essentially assisted by gravity and to some extent the draw that's produced by the expanding of the chambers. The semilunar valves at this point are closed. The atrioventricular valves are open. The atria then contract and force blood into the ventricles. In ventricular systole, the blood pressure builds before the ventricle contracts. The atrioventricular valves close, and this causes the first heart sound, which we designate as lub. The semilunar valves then open as the blood pushes against them. The blood travels out of the ventricles through the pulmonary trunk and the aorta, and the atria relax. In early diastole, at the end of systole, all four valves are briefly closed at the same time. The second heart sound is heard as the semilunar valves close, causing the dup sound. The atria finish refilling as pressure in the heart drops, and ventricular pressure is low, causing the atrioventricular valves to open back up. The cardiac output is the amount of blood that's pumped by each side of the heart in a single minute, and it's the ventricles that we look at primarily. The stroke volume is the volume of blood that's pumped by each ventricle in a single contraction, so each heartbeat is going to have a particular stroke volume, and this usually remains relatively constant. About 70 milliliters of blood is pumped out of the left ventricle with each heartbeat. Your heart rate is typically about 75 beats per minute. Remember, again, that the volume of blood that's ejected from the right side of the heart and the left side of the heart is going to always be the same. Your cardiac output is equal to your heart rate times your stroke volume, and so if we take those typical values and substitute them into the equation, what we find is a cardiac output a little over 5 liters a minute. Starling's law of the heart states that the more the cardiac muscle is stretched, the stronger the contraction will be. Changing the heart rate is the most common way to change cardiac output. Basically, the more we exercise, the greater our cardiac output is going to be, so that we can meet the oxygen demands of our tissues as their activity levels go up. By the same token, as our activity levels decline, our heart rate will decline down to a minimal level. And so what you're looking at here are some of the factors that can serve to increase heart rate. Innervation by the sympathetic nervous system, or the anticipation or the belief that there's a crisis can cause the cardiac output to go up, as can low blood pressure. Again, you don't want your blood pressure to get too high or too low. If it did, what would happen is that we would have cardiac insufficiency in the case of low blood pressure, not delivering enough oxygen nutrients to tissues to keep them alive. If we have blood pressure that gets too high, what's going to happen is we could run the risk of rupturing the most delicate vessels in the body, those being the capillaries. So the body's always going to try and keep your blood pressure within certain tight limits, and part of that is due to the control it exerts on the cardiac output. Other hormones that can control heart rate include epinephrine and thyroxine, both of which are going to tend to increase cardiac output, as will exercise. A decrease in blood volume will tend to accelerate cardiac output. A decreased heart rate is generally due to innervation by the parasympathetic division of the autonomic nervous system, as well as high blood pressure or blood volume, or decreased venous return. All of these will result in a decrease in cardiac output. Now, one of the things that we have to consider when we look at the cardiovascular system are the vessels through which the blood travels. And essentially, you've got the arterial system. This is the trip that the blood takes away from the heart, and the venous system, which is the trip that the blood takes back towards the heart. These vessels allow the blood to move around the body to the tissues and back. Remember that arteries will carry blood away from the heart. These consist of arteries and arterioles. Exchange between the tissues and the blood takes place in the capillary beds because of their thin walls. And then, as the nutrient poor and oxygen poor blood re-enters, well, it doesn't re-enter circulation, but as it continues its route around the body, it returns back towards the heart through venules, eventually emptying into veins, which eventually returns the blood all the way back to the heart. You can see here, if we look at the anatomy of a typical artery and a vein, that there's a lot of common features, but there's also some unique features that separate the two. They all have the same three layers called tunics. The tunica intima being that layer of tissue that's going to come into contact with the blood that flows through the opening through the vessel called the lumen, and it's actually coated with a special molecule called prostacyclin, which repels the formed elements of blood so that they don't stick to the lining of the blood vessel. And then you've got the tunica media, which is made up primarily of smooth muscle, which is capable of contracting or relaxing. And what that does is helps control blood pressure, but it also helps to redirect blood to areas that the body wants it to go. Okay, so you can see the tunica media here in the arterial circuit and here in the venous return. And then you've got the tunica externa, which is basically fibrous connective tissue which attaches the vessels to the rest of the tissue in the body. Now, one of the things that we have to keep in mind is that this smooth muscle activity is going to have forces that govern it. One of the forces that controls whether or not we see vasoconstriction or vasodilation is oxygen supply. In systemic tissues, the lower the oxygen concentration, the more the smooth muscle is going to dilate. The higher the oxygen concentration, the more the smooth muscle is going to constrict. There's also innervation that comes from the sympathetic division of the autonomic nervous system that causes the smooth muscle to constrict, and that results in an increase in blood pressure. Okay, and also there's the phenomenon of tissue injury, in which components are released from damaged tissue, local effects that cause the smooth muscle to vasoconstrict and control the loss of blood through an injury site. Arteries have a thicker tunica media than the veins do. The capillaries are the most delicate vessel in the body. They have only a single cell layer, a tunica intima, which allows for the exchange of gases and nutrients and waste products between the blood and the tissue. The veins have a thinner tunica media than the arteries do, and they also have valves that prevent the backflow of blood. Remember that one of the challenges with the venous return back to the heart is this idea that we have to fight gravity in order to get the blood back into the atria from areas that are below the heart. And without these valves to ensure the one-way flow of blood, that'd be very difficult to do. Also, the lumen of the veins is much larger than the arteries. So generally what we find in the venous system is large volumes of blood that are present at much lower pressures than they are in the arterial system. Okay, so arterial system, smaller quantities of blood at higher pressure with higher oxygen and nutrient concentration, and in the venous system, larger quantities of blood present in the venous system at lower pressures with lower oxygen and nutrient concentration. And here you can see in this next panel how it is that the skeletal muscle pump helps to ensure venous return to the heart. Remember that veins have a thicker tunica media and operate under low pressure and have a larger lumen than the arteries do, so to help in moving blood back to the heart, the larger veins use valves to prevent backflow and also skeletal muscle, which milks the blood in the veins coming towards the heart. Another phenomenon that helps the blood flow back towards the heart through the venous system is what we call the thoracic pump. This idea that you're constantly breathing produces pressure on the great vessels, the vena cava, that lead back into the heart, and this also helps to milk blood back to the heart. And there's also a little bit of a draw as you get nearer the heart in the venous system that will tend to suck the blood back into the chambers as they go into diastole. So all of those help, but essentially the bottom line is the more active you are, the more blood is going to come back to the heart, and the result is that the cardiac output will increase, and the blood will essentially circulate around the body with a much greater velocity. Most arterial blood is pumped by the heart, and the veins use the milking action of muscles to help move the blood. So huge differences there between the two systems. Also, keep in mind that when we consider the arterial system, that usually, with just a very few exceptions, arteries are much deeper inside the body than veins are. Veins being close to the surface means that if you have an injury such as a laceration or a puncture, the blood that you lose through a vein is going to come out under lower pressure and is also going to have a lower oxygen and nutrient content. So it's going to be easier to stop that flow of blood when the blood clots. The exception to the rule that veins are more superficial than arteries happens in the pulse points of the body, such as in the carotid arteries by the neck, in veins near the wrist, by the elbow and behind the knee, in the groin, and so on. And those are danger areas. If you do get a puncture or a nick there and you open up an artery, you'll lose massive amounts of blood very, very quickly. So again, it's a good design feature of the body that the veins tend to be closer to the surface. Capillary beds, which are pictured here, are made up of two types of vessels. A vascular shunt is a vessel that directly connects an arteriole to a venule. True capillaries are what we call exchange vessels, and this is where we swap oxygen for carbon dioxide and nutrients for waste products. Oxygen and carbon dioxide diffuse across these vessels due to their relatively thin walls. And remember that the direction of the diffusion is governed by the concentration gradient. In the tissues of the body that are supplied by capillary beds, what happens is that the oxygen concentration outside the capillaries is relatively low because the oxygen is constantly being consumed in metabolism, while the carbon dioxide concentration is relatively high because that's also an end product of metabolism. So when oxygen-rich blood gets to the tissues, what will happen is that the carbon dioxide will diffuse into the blood at the same time that the oxygen in the blood diffuses into the surrounding tissue. And the result then is that as the blood leaves the capillary bed and heads back towards the venous return, it's going to have a lower oxygen and higher carbon dioxide content, while at the same time those tissues that are being perfused by that blood that's coming into the capillary bed are going to have their oxygen and nutrient needs met. So here you can see what happens when we have a capillary bed that's fully perfused. There are areas of smooth muscle called sphincters that open up under low oxygen conditions and allow these blood vessels to fill up, and this assists with gas exchange and nutrient delivery. And then when oxygen conditions are met, these sphincters will then choke back down, and the blood will be routed from an artery back directly into a vein and then back towards the heart. Um, Keep in mind also that in the body, only a fraction of the capillary beds are open at any one time as a way to try and control blood pressure. In the event that we were to experience massive amounts of vasodilation, where every capillary bed would open up, the blood pressure would drop to the point where we would probably lose consciousness. Okay, so this is again controlled by the sympathetic division of the autonomic nervous system. Now, addressing the aorta, this is the largest artery in the body, and it leaves from the left ventricle of the heart. And it has several regions. The ascending aorta directly leaves the left ventricle, while the arch arches to the left, and down behind the heart it becomes the thoracic aorta as it travels through the thorax. Once it pierces the diaphragm and enters the abdominal pelvic cavity, it's termed the abdominal aorta, a branch of the descending aorta. The right and left coronary arteries that branch off the base of the aorta, remember, actually supply the heart muscle itself with its own blood supply. Okay, so ascending aorta, aortic arch, and then behind here, we can draw it in dotted lines. Let's do that in yellow. Would be the thoracic aorta, okay, as we drop down through here, okay, behind the heart, giving off branches as it descends, eventually going through the diaphragm and down into the abdominal region. Okay, so I'm just tracing in there for you the thoracic aorta, okay, and then the diaphragm would be approximately here, okay, lie right about there, and then below this region, and we'll draw that in, let's draw it in red, would be the abdominal aorta coming down here. Okay, so that's how the aorta travels through the body. The arterial branches of the aorta, of the aortic arch, are the brachiocephalic trunk. It splits into the right common carotid artery and the right subclavian, and the left common carotid artery that splits into the left internal and external carotid arteries, and then finally the left subclavian artery that branches into the vertebral artery. In the axilla, the region under the arms, the subclavian artery becomes the axillary artery, then branches into the brachial artery, then branches into the radial, median, and the ulnar arteries. The arterial branches of the thoracic aorta include the intercostal arteries, which supply the muscles of the thorax wall. Remember that intercostal basically indicates running between the ribs. Other branches of the thoracic aorta supply the lungs, which include the bronchial arteries, the esophagus, which is supplied by the esophageal arteries, and the diaphragm, which is supplied by the phrenic artery. In the abdominal aorta, the celiac trunk is the first branch of the abdominal aorta after the phrenic artery. The three branches of the celiac trunk feed the main organs that lie immediately under the diaphragm. They include the left gastric artery, which supplies the stomach, the splenic artery, which supplies the spleen, and the common hepatic artery, which of course supplies the liver. The superior mesenteric artery supplies most of the small intestine and the first half of the large intestine. The reason it's termed mesenteric is because the connective tissue that holds the digestive system against the body wall is called mesentery. In the abdominal aorta, we also have the left and right renal arteries, which supply the kidneys, the left and right gonadal arteries, which supply the testes in the male and the ovaries in the female, and the lumbar arteries, which supply the muscles of the abdomen and also the trunk. We should also mention that there are a couple of adrenal arteries, which supply the adrenal glands. The inferior mesenteric artery serves the second half of the large intestine. The left and right common iliac arteries are the final branches of the aorta, and they get their name from the fact that they run next to one of the major parts of the os coxa, which is the ilium, the big fan-shaped crested portion of the hip bone. The internal iliac arteries serve the pelvic organs, while the external iliac arteries eventually leave the body cavity and enter the thigh, where they become now the femoral artery. They run behind the knee, where they're called the popliteal artery, and then they branch from there to form the anterior and posterior tibial arteries that run on the front and the back of the tibia respectively. And here you can see the major arteries of the body. And again, the best way to remember these arteries and where they go is to remember the different parts of the body. So if we start up near the aorta, we can see the brachiocephalic artery. Again, it's called brachiocephalic because it's going to branch into an artery that supplies the brachial region, which is the axillary, which then becomes the brachial artery. And the cephalic portion of brachiocephalic indicates a branch that will eventually supply blood to the brain in the form of the internal and external carotid arteries up here in the neck. As we move down, you can see the thoracic aorta behind the heart here, and then as we run through the diaphragm, we become the abdominal aorta, where you can see the celiac trunk, where you would have now the splenic, the left gastric, and the hepatic artery branching off the celiac trunk. You would have the adrenal arteries feeding each of the adrenal glands, the renal arteries feeding each kidney. Then you've got the superior mesenteric artery, and if you look very carefully, we should be able to pick that up. It would come up right about here, okay? The superior mesenteric, and then underneath would be the inferior mesenteric, and then of course you'd have the gonadal arteries, which would supply now the testes in the male and the ovaries in the female. Those are also a pair, okay? They occur as a pair of arteries. And down here, of course, where we split now into the right and left common iliac, we will then split again into the internal and the external iliac, and the minute we leave the abdominal pelvic cavity, it's referred to as the femoral artery. You can see here, okay, and you can also pick up behind the knee where it becomes the popliteal, which then branches into the anterior and posterior tibial arteries, and then all the way down here on the top of the foot, it's termed the dorsalis pedis, and this happens to be one of the places in the body where blood flows extremely close to the surface in an artery, so it's a good place to take a pulse, in addition to behind the knee, in the groin, in the region up here by the neck, and of course down here by the wrist, okay, just to name a few of the major pulse points in the body. The superior and inferior vena cava enter the right atrium of the heart. The superior vena cava, of course, draining blood from areas above the heart, while the inferior vena cava drains structures below the heart, and as a result, getting blood into the inferior vena cava requires that we fight gravity. And this is where the skeletal muscle pump and the thoracic pump, when you breathe, play their major role in getting blood back up to the heart in order to maintain cardiac output. The veins that drain into the superior vena cava include the radial and ulnar veins, which drain into the brachial veins, which drain into the axillary vein. This drains the arms. The cephalic vein drains the lateral aspect of the arm and empties into the axillary vein, which runs under the arm in the armpit area. And the basilic vein drains the medial aspect of the arm and empties into the brachial vein. The basilic and the cephalic veins are jointed at the median cubital vein, which is near the elbow. In the superior vena cava, the subclavian receives venous blood from the arm through the axillary vein and venous blood from the skin and muscles through the external jugular. The vertebral vein drains the posterior part of the head, and the internal jugular vein drains the dural sinuses of the brain. And this is why the neck is an extremely vulnerable region if you were to get an injury, because we'd run the risk of severing the jugular vein and or the carotid artery. The left and right brachiocephalic veins receive venous blood from the subclavian veins and vertebral veins, which run on either side of the vertebral column in the cervical region, as well as the internal jugular veins. The brachiocephalic veins join to form the superior vena cava, which empties into the right atrium of the heart, while the azygous vein drains the thorax. It's located on the posterior body wall near the vertebral column. The veins that drain into the inferior vena cava include the anterior and posterior tibial veins and the fibular veins, which drain the legs. Those are sometimes also called the peroneal veins. The posterior tibial vein drains into the popliteal vein, which drains into the femoral vein, which drains into the external iliac vein. The great saphenous veins, which are the longest veins of the body, receive the superficial drainage of the legs. Each common iliac is formed by the union of the internal and external iliac vein on its respective side, which then drains into the abdominal vena cava, which is basically the distal portion of the inferior vena cava. Veins that drain into the inferior vena cava also include the right and left gonadal veins, which drain the right ovary in the females and the right testicle in males, and the left gonadal vein, which empties into the left renal vein. The left and right renal veins drain the kidneys, and the hepatic portal vein drains the digestive organs and travels through the liver before it enters the systemic circulation. And it does so as the hepatic vein, which basically comes out of the top of the liver and goes directly into the inferior vena cava. Veins that also drain into the inferior vena cava include the left and right hepatic veins, which drain the liver. And here you can see the distribution of the veins in the body. Okay, so again, we start up near the top. The dural venous sinuses are going to drain into the external internal jugulars, which are going to drain into the brachiocephalic trunk, which drains into the superior vena cava. You can see here the radial, the median, and the ulnar veins joining up here with the antecubital vein and eventually draining in along with the cephalic and the basilic vein into the axillary vein, which is going to drain into the brachiocephalic vein, which is going to drain into the superior vena cava. Okay, the same is true basically on the other side of the body. Again, you've got the brachiocephalic vein, which is going to drain the left half of the body from the axillary, antebrachial, and brachial regions, and enter now the superior vena cava. As we leave the heart, as we head down to the legs, we can work our way up. Again, the dorsalis, the dorsal venous arch is going to drain into the anterior tibial vein. The posterior tibial vein will join that, forming the popliteal vein, which will drain into the femoral vein. The femoral vein, along with the great saphenous, is going to drain into the iliac vein. Okay, you can see the iliac vein right here next to the ilium. And then the iliac veins will fuse. The external iliac vein and internal iliac vein will form the common iliac vein, and the common iliac veins will drain into the inferior vena cava, which will head back up to the heart. Other veins that drain in along here include the superior mesenteric and the renal and hepatic portal, splenic, and hepatic veins. All of these, again, under the diaphragm, and of course the inferior vena cava eventually makes it back up to the right atrium of the heart. If we look at the arterial supply, the internal carotid arteries divide into the anterior and middle cerebral arteries, which supply most of the cerebrum, while the vertebral arteries join once within the skull and form the basilar artery, which serves the brainstem and the cerebellum. The posterior cerebral arteries form the division of the basilar arteries, which supply the posterior cerebrum. And so if you look at the underside of the brain, you can see these very important structures. So you can see the anterior and posterior blood supplies united by communicating arteries, resulting in the formation of what we call the circle of Willis, which is essentially a type of circulation that we call an anastomosis. Essentially, collateral circulation. It's basically to ensure that if we have a block in some other major vessel in the body, that blood can bypass this block and still perfuse portions of the brain without being completely shut off. So we can see here the vertebral arteries joining into the basilar artery that runs right across the pons right here. Okay, this is the medulla oblongata, this is the pons, and then feeding directly into the posterior cerebral artery, which joins the circle of Willis. These are the posterior communicating arteries. These are the anterior cerebral artery here, okay, coming out here, and then you've got the anterior communicating artery right up front, okay, and that completes the circle of Willis. This basically redundant method of ensuring that we supply blood to all parts of the brain, even in the event that we have a stroke. That's what we mean by collateral circulation. You can see here the internal carotid, where it feeds into the circle of Willis as well, okay, right here and here. And you can also pick up the middle cerebrals. Okay, this is looking underneath the temporal lobe here. You can see the blood supply coming through here, and we also need to make note of the let's see here, the posterior cerebral artery, which we noted earlier, and that covers essentially the collateral circulation to the brain. Now, when we're in our mother's womb, we have a particular challenge, and that challenge is to supply blood to the developing fetus, even though the respiratory system isn't yet engaging in gas exchange. So the fetus receives and exchanges gases, nutrients, and wastes through the placenta. The umbilical cord contains three vessels: the umbilical vein, which carries blood that's rich in nutrients and oxygen to the fetus, and the umbilical arteries, which carry carbon dioxide and debris-laden blood from the fetus to the placenta. Blood flow bypasses the liver through the ductus venosus, and it enters the inferior vena cava near the right atrium of the heart. The blood flow bypasses the lungs, and blood entering the right atrium is shunted directly into the left atrium through the foramen ovale, while the ductus arteriosus connects the aorta and pulmonary trunk in order to ensure that oxygenated blood gets to both the pulmonary and systemic circuit. It eventually becomes the ligamentum venosum at birth as it collapses due to pressure changes. So if we if we trace the flow here, okay, we'll do this with a black pen. All right, let's take the blood that's coming back to the baby from the placenta. Okay, it's picked up its oxygen from the mother's blood supply as well as its nutrients, and it has basically relieved itself of carbon dioxide and other waste products. And so the blood flows back to the fetus through the umbilical vein, okay, coming up through here, and eventually what happens is that we're going to pass under the liver, okay, this is the ductus venosus, and then we're going to enter right up here the inferior vena cava. From here, the blood is going to enter the right atrium, along with blood that's draining from areas above the heart through the superior vena cava. And then what will happen is that the blood will flow through the tricuspid valve shown here into the right ventricle. At the same time, because there is a hole called the foramen ovale between the right and left atrium, as the right atrium squeezes, blood will also be forced into the left atrium. Okay, so you've got two places for the blood to go here. You've got the right ventricle, and you've also got the left atrium. Then, as the blood pours from the atria into the ventricles, okay, you can see that here, what will happen is that the ventricles will begin to squeeze, these valves will shut, and then the aortic and semilunar valves will open up. The blood will pass into the aorta, okay, you can see that blood supply here going into the aorta, but it so happens that the aorta and the pulmonary trunk are joined by the ductus arteriosus right here. And so what happens is that as oxygenated blood enters the aorta, it also enters the pulmonary trunk because the two are joined by this bypass. As a result, both the lungs here and here are going to receive oxygen-rich blood as well as the rest of the body via the aorta. So what will happen eventually is that this blood will flow to the tissues. It'll become exhausted of its oxygen nutrient content, and then this oxygen and nutrient poor blood will exit the fetal circulation in the body, and it's going to enter now the umbilical veins, okay, which are going to, the umbilical arteries, which are going to drain deoxygenated blood away via the internal iliacs, okay, you can see them here, back into now the umbilical artery proper, where it will wrap its way around, okay, and eventually this low oxygen blood will reach the placenta, and at the placenta what will happen is that carbon dioxide will be exchanged for oxygen at the same time as waste products are exchanged for nutrients, and then the oxygen-rich, nutrient-rich blood will flow back to the baby via the umbilical vein, and the entire process will repeat itself. And that's essentially a way to continue to get oxygen to both the pulmonary and the systemic circuit while the baby's still inside the womb, because you can't use the lungs to get oxygen at this point yet. And so you can see here in this key the different oxygen concentrations in the different vessels. Okay, high oxygen concentration indicated here as red, moderate oxygenation here as sort of purple, low oxygen in pink, and very low oxygen here in blue. This is just a look again at the heart, and it's primarily here to show you the chambers and the valves that connect them, and also um, where the ligamentum arteriosum would be located, which would be up here right between the aorta and the pulmonary trunk. It collapses due to the pressure changes that the circulatory system experiences when the baby takes its first breath after it passes through the birth canal. Now, there are some special circulatory routes in the body that we need to be aware of, and these include the hepatic portal circulation, which drain the digestive organs, the spleen, and the pancreas. The hepatic portal vein carries nutrient-rich blood to the liver from the digestive system, and the liver helps to maintain a proper glucose, fat, and protein concentration in the blood. The major vessels of the hepatic portal circulation include the inferior and superior mesenteric veins, the splenic, and the left gastric veins. And again, all of these eventually drain into the liver. The liver then adjusts the blood chemistry by determining either to store nutrients for later use in the form of fat or as glycogen, or to pass the nutrients on to the rest of the body in the event that the metabolic needs are very high at that point. So you could think of the liver in this role as the body's kitchen, essentially preparing supplies for the rest of the body to eat if needed. Other places where you find portal circulation systems in the body where the blood actually passes through two capillary beds before it re-enters the venous circulation include the hypophyseal portal system that exists between the hypothalamus and the anterior pituitary, just to name another example of this exact same kind of setup. Now, we've talked about pulse points before. They happen to be places in the body where arteries are very, very close to the surface. A pulse is basically a pressure wave of the blood, and it's monitored at special points in the body called pressure points, where you can easily palpate an artery because there's not a lot of overlying fat. Pulse averages about 70 to 76 beats per minute at rest, and you can see all the different places where you can take a pulse: on top of the foot, on the inside of the ankle near the medial malleolus, behind the knee in the popliteal region, near the femoral artery in the groin, the radial artery and the brachial artery in the wrist and the elbow respectively, the common carotid and facial arteries, as well as the superficial temple. And again, these are places where arteries are close to the surface and there's not a lot of fat covering them up, so it's easy for us to pick up those pressure waves as they change. The reason that we can't take a pulse in a vein is because the pressures are too low. We wouldn't be able to detect them, and you can see that in this diagram here, showing you the changes in overall pressure as we head away from the heart, as well as the changes in pressure differential as we head away from the heart. Measurements by health professionals are made on the pressure that's in the large arteries. The systolic is the pressure at the peak of ventricular contraction, while the diastolic is the pressure that occurs when the ventricles relax. We generally write blood pressure as systolic over diastolic, and so a typical reading might be 120 over 80 millimeters of mercury. Remember that 760 millimeters of mercury is a single atmosphere, and so we use this as a way to measure blood pressure using a device called a sphygmomanometer, which is essentially a pressurized cuff that can sense changes in blood pressure, indicating the pressures on a dial, and we usually use a stethoscope in order to determine when the systolic and the diastolic pressure are ascertained. Pressure in blood vessels decreases as distance from the heart increases, and you can see that here as we head away from the heart via the aorta, hitting the arteries and then the arterioles, and then we have this dramatic drop that we experience from the arterioles to the capillaries because of the narrowness of the vessels and also the distance from the heart And so by the time we reach the venous return, the pressures are so low that they wouldn't be detectable with a sphygmomanometer, and the use of the sphygmomanometer is seen on this next page here. So note figure 1121 in your book, but what I'm going to show you here in just a moment is a procedure for measuring blood pressure using a sphygmomanometer that'll detail all of this. So we'll flip through this real quick, and then what I want you to do is listen to this brief video that describes the proper use of a sphygmomanometer.
Before you begin, be sure to note whether the patient is sitting, standing, or laying down during the pressure reading. The standard position is with the patient sitting, with the patient's arm relaxed. However, this position is not always practical in an emergency situation. First, choose the correct sized cuff for the patient's arm. Put the cuff around the upper arm, with the cuff mark at the medial side of the biceps brachii, about an inch above the anterior elbow. Next, with your first two fingers, feel or palpate for a pulse on the radial artery. The radial pulse is found on the thumb side of the forearm at the wrist. Now, squeeze the bulb to inflate the cuff until the pulse can no longer be felt. The pressure in the cuff matches the pressure needed to close the vessel and to stop the flow of blood. Keep inflating for about 30 millimeters of mercury above the point where no pulse is felt. Place the stethoscope just under the distal part of the cuff over the brachial artery. Slowly release the pressure at about 2 millimeters per second. As the pressure in the cuff drops, blood begins to pulse through the vessel. When you hear 2 consecutive beats, record this pressure reading as the systolic pressure. Keep releasing the pressure slowly until you can no longer hear the sounds in the stethoscope. The cuff pressure reading at the time when the sound disappears from the stethoscope is called the diastolic pressure. This represents the pressure exerted on the walls of the arteries when the heart is at rest. Open the valve to deflate the cuff completely, but leave the cuff in place to save time for sequential readings. Record your information as soon as possible to avoid forgetting it. Be sure to repeat the readings over time to check for changes in blood pressure as the patient progresses. The inflated cuff compresses the tissues and vessels to stop the arterial pulse. As the air is released from the cuff, the blood vessel opens just enough to allow the high-pressure blood to pass through the vessel. As this blood passes, the partially compressed cuff creates turbulent blood flow. This turbulence can be heard by placing a stethoscope under the distal end of the cuff. At this pressure, we are reading the systolic pressure. As you slowly deflate the cuff, there is less compression of the vessels, and the blood flow becomes less turbulent, so the sound is no longer heard. The diastolic pressure represents the pressure exerted on the walls of the arteries when the heart is at rest. Remember to fully deflate the cuff when finished.
BP refers to systemic blood pressure, which is going to be affected by age, weight, the time of the day, your activity level, the position of your body, and your emotional state. So there's a lot of things that can cause transient changes in your blood pressure. Cardiac output is the amount of blood that's pumped out of the left ventricle per minute, while PR stands for peripheral resistance, or the amount of friction that the blood encounters as it flows through the vessels. And this is going to be a force that's exerted in the opposite direction to the flow of blood. The narrowing of blood vessels and increased blood volume are going to increase peripheral resistance, while a widening of the blood vessels and decreased blood volume are going to decrease peripheral resistance. So we can basically use a mathematical equation to express blood pressure as equal to cardiac output times peripheral resistance. And again, blood will always flow from a region of high pressure to a region of low pressure. And the difference in the two pressures is going to be responsible for the speed with which the blood flows from one area of the circulatory system to the other. So what are some of the other factors that influence blood pressure? Well, there is a neural influence on blood pressure. The autonomic nervous system can make adjustments via the sympathetic division to the diameter of the arteries. Well, there are also organs that come into play. The kidneys, for instance, can regulate blood pressure by controlling blood volume using a couple of different hormones. Antidiuretic hormone, which increases water retention by the kidneys by acting on the distal convoluted tubule in the collecting duct, and aldosterone, which causes increased sodium reabsorption by the kidneys, which in turn leads to increased water retention. And both of those are going to end up increasing blood volume and blood pressure. Renin is also another hormone control over blood pressure. It eventually results in the formation of angiotensin II, which is a potent vasoconstrictor, and it also stimulates the production of aldosterone. Temperature also has a major role. Heat has a vasodilating effect, so pressure tends to decrease under the effects of heat, while cold has a vasoconstricting effect. And there are also various substances that can cause an increase or decrease in blood pressure. These chemicals can be things such as diuretics, which tend to cause you to lose blood volume and blood pressure. You also have chemicals that act on the cardiac output. Okay, they can adjust the force of contraction of the heart, and that can also change the pressure in the vessels. And also your diet can have a major influence on your overall blood pressure. And remember that it's bad for your overall health to have blood pressure that's consistently high or consistently low, because in the event that you're, say, hypertensive all the time, you're eventually going to damage capillaries. In the scenario of having low blood pressure, the pressure could get to the point where you don't adequately perfuse the organs, and they lose out on the amount of oxygen and nutrients they need to sustain life. The normal human range is highly variable. It can run from 140 to 110 millimeters of mercury systolic to between 80 and 75 diastolic. Hypotension is defined as a low systolic pressure, generally below 110 millimeters of mercury, and it's often associated with illness, while hypertension is defined as a high systolic pressure greater than 140 and can be dangerous if it is chronic. Now, we can always have transient increases in our blood pressure, which can come as a result of any of the factors that we've cited earlier. It's only if these pressures stay consistently high over an extended period and in a variety of activity levels that we worry about the effect on overall health. The same is true of hypotension. Now, in the capillaries, what's happening is that we're actually supplying nutrients and oxygen to the cells that need them to live while removing waste products. And this is all facilitated by the presence of a concentration gradient at the tissues that favors the movement of carbon dioxide into the blood and oxygen moving out of the blood, while waste products simultaneously move into the blood and nutrients move out. And here you can see an example of what's happening at the level of the plasma membrane in order to facilitate this type of movement. Direct diffusion across plasma membranes is driven entirely by differences in concentration on the different sides of the cell membrane. Active transport can also play a role. Endocytosis and exocytosis can help to move substances across the cell membrane as well. Also note that some capillaries have physical gaps in them called intracellular clefts, and the plasma membranes are not joined together by tight junctions in capillaries. There are also pores in certain capillaries, such as those that are found in the glomeruli of the kidneys, that allow substances below a certain molecular weight to easily pass through. And so this diagram is showing you the different methods by which substances move in and out of the blood. Okay, direct diffusion, which is a major force in the exchange that takes place in the tissues. You can also have diffusion through intracellular clefts, through pores, and of course active transport as well. So if we're looking here at capillary beds at the levels of the tissues, what we find is that blood pressure tends to force fluid and solutes out of capillaries at the arterial end of the capillary bed. The osmotic pressure draws fluid into the capillaries. The blood pressure is higher than the osmotic pressure at the arterial end, while it's lower than the osmotic pressure at the venous end of the capillary bed. And so what we have here when the blood first enters is a net movement of fluid out of the capillaries and a net movement of nutrients and oxygen out of the capillaries, while at the same time we have a net movement at the venous end of the capillaries of carbon dioxide and waste products into the blood, and as a result what happens is that the pressure begins to drop and the amount of waste products in the blood begins to rise, the nutrient content begins to fall, and eventually the deoxygenated blood is going to flow back to the heart through the venous return. So you can see here the difference in blood pressure at the beginning of the capillary bed and at the end of the capillary bed. Now, one of the things that we should talk about before we finish this chapter is how the circulatory system actually forms. And it's quite a fascinating story because when the circulatory system first begins to generate discernible structures, what we find is a very simple pair of beating tubes that develops in the embryo and begins pumping by the 4th week. These beating tubes will then fuse with each other and eventually form first a 2-chambered, then a 2.5-chambered, then a fully 4-chambered organ by the end of 7 weeks. Very few structural changes occur after the 7th week. The reason that this is an interesting series of events is because if you look at the hearts of simple organisms and move up the evolutionary ladder, what you find is a progression very similar to what we see in the development of the human heart. For instance, if you look at the heart of an earthworm, it's simply a beating tube, while the heart of a fish is a 2-chambered heart, the heart of an amphibian a 2.5-chambered heart, as is that of a reptile, which you could almost consider a 3-chambered heart, and finally, by the time we get to the mammal, okay, which is more closely related to the human being, what we find is a fully functioning 4-chambered heart. And so this seems to follow a philosophy in biology that that basically states that it's entirely highly likely that the events that happen during human development seem to mimic the events that have happened during the course of evolution. As a result, we often state that ontogeny recapitulates phylogeny, which is simply a restatement of that particular set of assertions. Aging problems associated with the cardiovascular system include venous valves, which begin to weaken, leading to varicose veins, and progressive arteriosclerosis, where deposits tend to narrow the lumen of the vessel and also reduce its flexibility, and as a result lead to a hypertensive condition. The loss of elasticity contributes to this. Coronary artery disease often results from the vessels getting filled with fatty calcified deposits. If the vessel can't distend, it can't stretch, and it can't relax with the beating action of the heart, what happens is that the pressure against the walls slowly begins to increase, and over time, that steady increase in blood pressure can lead to the rupturing of capillary beds. It's also important to note that as the lumens of the vessels narrow as a result of these deposits building up, that we tend to see um, the formation of emboli more frequently. These traveling clots can eventually lodge in a capillary bed and choke off oxygen supply to downstream tissues, and so we see an increased risk of stroke and myocardial infarction as a result of this as well. Okay, please make sure and review this material as it will be covered on the upcoming quizzes and exams, and I will see everybody in class.